treatments and the immunotherapy. Okay. As I've had for all mine slides so far, for each module, you do have a list of learning objectives, a general list of learning objectives that is located in that overview. Okay, so we're going to be looking at differentiation between innate immunity and adaptive immunity. We're going to be looking at the role that your normal microbiota play in your innate immunity, identifying the physical and chemical barriers that comprise the first line of defense. So basically talking about the first line and recognizing those aspects of the first line of defense, followed by recognizing all the components of the second line of defense, which are both the first and second line of defense are all part of that innate immunity. And then recognizing the role of phagocytosis, inflammation, complement, interferon, fever, all of which are part of that second line of defense. So kind of being able to walk through those different processes and how they work. Some of them are, what specifically do they target in helping us eliminate the pathogen from our system. So that being said, okay, we do have three main lines of defense. We have our first and second line of defense, which fall under what we refer to as the innate immunity. The innate immunity consists of external and physical barriers, right, that prevent entrance, usually associated with that first line of defense, stopping the pathogen from even entering into the body. And then we have, for the part of that second line of defense, is these pathogens have gained entry. What do we have put into place? We have we have cells that are going to be involved in the process of that second line of defense. We're going to have bloodborne chemicals and a general process, okay, that involves the combination of those cells and the chemicals in that second line of defense, working in a manner to eliminate this pathogen. In the case of innate immunity, it does not improve upon repeat exposure. So it doesn't matter how many times you come in contact with a particular pathogen, your response to that pathogen with innate immunity is going to be the same. Okay. It is also, with innate immunity, is not directed against, it is directed against a variety of pathogens, not in a specific pathogen. Okay. So what we're going to be looking at is that the innate immunity is going to be recognizing, say, these protective cells are going to be recognizing these particular pathogen-associated molecular patterns that many different pathogens will have, right? And so it doesn't matter whether it's a gram-negative cell or a gram-positive cell. It doesn't matter whether it's fungi or bacteria, right? It doesn't, in the innate immunity, it doesn't distinguish that type. It doesn't separate. The response is going to be the same no matter what it is. As compared to the adaptive immunity, and the adaptive immunity we refer to as a third line of defense. And in this case, we're looking at, in the adaptive immunity, we are also looking at these protective cells. Just like with the second line of defense, these protective cells are going to be coming from the blood, and these cells are going to have specific activities. These are our lymphocytes. They are going to be cells that we can produce memory. So we have this clonal ability and memory ability with these cells in order to get a better response each time you come in contact with exposure. So your first involvement with that particular pathogen, exposure to that pathogen, might be a delayed response, but on secondary interactions or any interactions after that first, you will have a stronger response and quicker response due to that memory that does exist for that third line of defense. The adaptive immunity is broken into two big categories. We have our humoral immunity, also known as the antibody immunity or antibody response. And this is going to involve the B cells. And then we have our cell-mediated immunity, which is gonna involve our T cells, okay? Now, the adaptive immunity side, that third line of defense, is going to be what we talk about in more detail in module 12. Okay, so we're not, in this module, we're putting those two aside. Or that one, excuse me, those two processes aside and fall into the third line of defense. So when you break it down, the portion that I have highlighted in yellow is that module 11 we're gonna be talking about for this particular module. And that's that innate, non-specific response, okay? Where we're gonna be looking at that first line of defense. The first line of defense is gonna consist of structures, physical barriers, or mechanical barriers that are going to prevent access to the sterile regions of the body, the regions of the body that need to stay sterile. And then we also have chemicals that are going to be involved in some processes that work to prevent those pathogens from even entering into the system. Okay? The second line of defense is upon entry, so now the pathogen has made entry, it's made it past the first line of defense, and we're now in the second line of defense. And the second line of defense consists of cellular and chemical systems that, or processes that are going to immediately play a role in trying to control that foreign body that has gained entry. The main ones that we're going to be talking about are the inflammatory response, interferons, phagocytosis, and complement. You can also include fever, is going to be discussed too, so that's another one that we'll cover. Okay? So that's module 11 that we're going to be covering. The third line of defense, as I already mentioned, third line of defense is going to be in module 12. That's that cell-mediated immunity, the humoral immunity or antibody response. In this case, it's a specific response, right? Uniquely developed uniquely for each microbe that you come into contact with, regardless as to it's specific to that particular microbe. In fact, the specificity is not just to the microbe, but actually to an antigen, a specific region, an epitope that's being recognized that particular pathogen produces. Now, the third line of defense, we will go into this more again, but it can be acquired through natural processes or artificially acquired. Okay, it can go either way. If you are looked at for both natural and artificial, there are passive processes and there are active processes. Okay, so just to break down that aspect, if it is naturally acquired but active, an active naturally acquired process, then we're really talking about interaction with the pathogen. Okay, if it is a passive naturally acquired process, an example of that would be maternal antibodies being received either in utero, passing across the placenta to the growing fetus, or through breast milk when breastfeeding the baby, okay, after birth. We have, for the artificially acquired, the active artificially acquired process would be through vaccinations, right? Because if you receive the vaccination, your body is going to actively be involved in giving a response. And then we have the passive artificially acquired process, and this is where you have, you receive, say, immune serum, so preformed antibodies that you're receiving. Okay, that's a passive process. There are all those antibodies already pre-made, and you're taking advantage of those. Okay, and so we'll touch upon all of that in again in module 12. All right, so second learning objective, of course, they've got listed was identifying the role of the microbiome in the innate immunity. So basically, your normal microbiota. What is its function? It actually is very important for our immune system. It has a role. Its role is microbial antagonism. Okay, so this isn't the first time I've used it. I've actually brought it up already in the last unit. And this is where the normal microbiota, those bacteria that you keep happy and healthy and provide nutrients for, and they provide nutrients for you. You have a relationship with them, right? They're your little babies that you take care of all the time, even if you don't want to believe that's the case. But what they're doing is they're as being established, they are competing, right, for real estate in your body and nutrients, right, attachment sites and nutrients. So they are going to be involved in consumption of nutrients, making it less accessible for pathogens. It's one way in which they're going to prevent these pathogens from establishing. They can sometimes create an environment that's unfavorable for other microorganisms. The text if you read through the text, you're gonna notice they talk about the fact that on the surface of the skin, there are some bacteria that live on the surface of the skin that actually have enzymes that utilize the sebum, the oils that your skin produce, right, and breaks it down. In the process of metabolizing it, it produces an end product that is slightly more acidic than the sebum is on its own, right? And that additional acidity inhibits some microorganisms, thus creating an antagonistic environment for pathogens to establish themselves. You have the fact that they are attached to the surfaces, making it more difficult for these pathogens to attach then to the surfaces because the real estate's already established as far as who's living where in that environment. The normal microbiota stimulates our second line of defense. So it stimulates some of our white blood cells that are monitoring the system. It keeps them occupied and active, which is a good thing. Therefore, they're more likely to then catch these pathogens before they establish. Some of the microorganisms will actually produce antimicrobial compounds that will prevent the growth of other organisms. And then we also have the fact that these normal microbiota are promoting our overall health, okay? So and in doing so, they keep our immune system strong, okay? So because when we're healthy, that's when we have a repressed immune system. When we're not healthy is when we have a repressed immune system. When we're healthy is when our immune system is healthy, is also. What they'll do in order to promote the health of ourselves would be the fact that they digest many of our foods and provide us those vitamins or precursors to the vitamins that we have to obtain from that food source. So they're providing it for us, such as the precursor for vitamin K that's important for blood clotting. Also, blood clotting is important for our second line of defense and immunity, right? Folic acid is what we get from these normal microbiota in our intestines. They are providing that. That's important for nitrogenous bases and nucleic acids. So that's going to be keeping our cells healthy, right? And vitamin B5 is another one, which is important for glucose metabolism. So we have a whole bunch of things that they're doing for our body that keeps us healthy, and in doing so, it is going to inhibit the growth of potential pathogens or the ability of those pathogens to establish. All right, so looking at our first line of defense, aside from the presence of these the normal microbiota in the environment where we have our first line of defense, what is our first line of defense? The biggest, of course, is the physical barriers that separate the internal sterile region of the body from the outside world. So we're already, discussed this and and everyone's familiar with the fact that, we have the skin is a major component, the obvious one that you can think of as far as an organ that is in direct contact with the outside environment, right? And the skin is composed of two major layers. We have the epidermis and the dermis. The epidermis has multiple layers of tightly packed cells, it's difficult to penetrate, right? We have shedding of the dead skin cells, so that's actually going to be brought up in the mechanical aspect also, is the fact that we're continuing to shed these dead skin cells. Therefore, any pathogens or microorganisms that we come into contact, if they're trying to attach to these cells, they're going to be removed, right, as the cells are shed. And then we do actually have epidermal dendritic cells, which have the ability to perform phagocytosis that can monitor this area. Okay, so it is monitored also by these phagocytic cells. The lower level of the layer of the skin, which is the dermis, That's the area that is known to be sterile. It doesn't have normal microbiota present. That region it has those collagen fibers that help resist abrasions, right? Keeping our skin, adding strength to our skin, as well as helping with the pliability of the skin. We'll also be talking about, in the next slide, I'll cover some of the chemical aspects that are going to be very important for this physical barrier in order to help maintain it. And then, of course, the next physical barrier would be the mucous membranes. The mucous membranes line then the respiratory system, digestive tract, the your reproductive system. And so it's basically that it's the mucous membrane is a little different than the skin, right? It's got layers to it also, an outermost layer, right? The epithelium layer. This epithelium layer, though, is thin. Sometimes even as thin as a single layer thick, but we do see shedding of cells from this, continual shedding and replacing of these cells, and so that prevents pathogens from being able to attach potentially if those cells get shed. Where if they attach, they happen to be cells that are going to be shed sooner, basically. We also have the fact that, again, these cells are tightly compact, right? So that's going to prevent entrance and the ability to cross into deeper tissue. We have dendritic cells also monitoring this outermost layer of the mucous membrane. And we have goblets and ciliated columnar cells that are involved with removing pathogens also. Okay, we'll talk about the most obvious chemical structure that's there, which is the mucus itself, too. Right, that'll be again touched upon in the next slide. The deeper connective tissue is going to be one which supports and maintains that outermost layer that has direct contact with the outside. Okay, and then lastly is our endothelia, which is the epithelial cells lining the urogenital tract, the blood vessels, the lymphatic vessels. It's what creates that blood-brain barrier, and so it too is a separation or division between the two different areas. So it'll have a kind of a lumen aspect, the inside or the tube versus the outside, right? Because it is possible that microorganisms can get into the blood, and if they get into the blood, we don't want them then transferring very easily out into the surrounding tissues. As far as mechanical defenses and chemical defenses, okay? For mechanical defenses, we've got, again, as I said, mechanical defenses would be shedding of the skin cells, the outermost skin cells, right? So that continual shedding process. That shedding process goes both for the skin as well as for the mucous membrane. But the mucous membrane also has the addition of the presence of the mucus itself and the expulsion of the mucus. The mucus is going to flow right from the nasal cavity to the pharynx, and then you swallow or be flushed up from the lower respiratory tract into the pharynx, and then you swallow that continually. So then it goes through the digestive system and then out the body. Okay. You have peristalsis, which is that wave-like contraction that is going to occur with the intestines to move the solid material through the intestines. Well, in the process, that solid material is rubbing up or scraping up against the epithelial cells of the mucous membrane, taking with it potentially the microorganisms. The solid material, of course, is being digested with the bacteria. So the bacteria, the bulk of your solid waste that you release out into the environment is actually bacteria-based. Okay, so bacterial cells that are working away at digesting that. So excretion of the feces, right? As far as voiding it for urine, that process is one in which you are having a large volume of liquid going through a very, very small tube. Okay, it's a flushing mechanism. okay? Because that liquid, as it pushes through that the urethra, it's moving the force of that is going to be pushing out any potential pathogens as they try to, if they're modal, as they try to move up the urethra. Okay, so that is another way in which we keep that region of the system clear. Okay, and then we have blinking, right? Blinking. So we have the lacrimal apparatus. This produces, the lacrimal apparatus produces the tears. The actual gland is on the upper outside of the eye, and in the fluid then washes down. You blink, that's like the windshield wipers, right? Every time you're blinking, that is it works in a similar fashion. It pushes the tear as well as moves across direct contact with the surface of the eye in order to wipe away any bacteria or debris, moving to the inside of the eye. That then deposits into the nasal cavity, where then, of course, it washes and mixes with, either it goes through the nose, out the nose, or down the pharynx, right, and into the stomach. So that process is a continual wiping process to ensure that we don't have entrance of microorganisms through that route. The tears themselves have lysozyme. Lysozyme is an enzyme that breaks down the cell wall, that peptidoglycan of the cell wall, bacterial cells. Okay. So that kind of moves us over to the chemical defenses, and so some of them I might have touched upon. Otherwise, we're going to go into a little more. So when you're talking about the skin, perspiration is going to be important. It has perspiration is secreted, of course, by the sweat glands. Within perspiration is salt, right? And the whole purpose of perspiration is for evaporation, cooling of the body. So what leaves? Evaporation of the water, right? And if salt is present, the salt is left behind, which is why the skin is salty when taste, right? If you taste, the skin is salty, and that is actually important, right? Because the presence of that salt means that it's going to inhibit many microorganisms, and many microorganisms cannot tolerate those higher levels of salt, which we already talked, kind of worked with it in the laboratory setting when we were, in order to try to isolate and select for Staphylococcus aureus, which is an organism found on the surface of the skin. We took advantage of that, right? It's an organism, if it grows on the surface of the skin, usually it's an organism that can tolerate higher levels of salt, so it's halotolerant, right? But there are a lot of other organisms out there that aren't going to be able to grow on it with that higher level, as we saw with things like E. coli, okay? Cannot grow at that higher level of salt. Perspiration also has antimicrobial peptides. We'll talk about that in a moment. I've got a couple slides demonstrating a couple examples. It also contains lysozyme. Okay, as I already mentioned, that is going to break down the peptidoglycan. It actually cleaves the amino acid bridge of the peptidoglycan, which then weakens that structure. Sebum, okay, is important, of course, for the skin also. It's secreted by the sebaceous glands, or the oil glands. It is going to help with keeping that skin pliable, okay? So it's less likely to break or tear. For individuals who don't have a lot of oil glands, you tend to see, especially in areas as like the knuckles, where you have bending occurring on a regular basis, or when you see drying out. I've got some sisters who would run into this problem when they live in colder weather, right? And the oil's not moving as well, and then you get cracking of the skin, the drying out of the skin, cracking of the skin, right? That's going to be a problem if your skin, the whole purpose of your skin is to prevent access to deeper tissue, right? Because cracks and breaking of the skin provides entry for the potential pathogens. The sebum also lowers the skin pH. That is going to make it more inhibitory for pathogens. You have lysozyme I already talked about. Lysozyme is in tears, it's in perspiration, it's in the saliva, it is, it's in various, it's in the mucus, so it's in various areas of the body that is going to play that role in breaking down the cell wall, okay? Stomach acid, okay, just the fact that the acidity itself is going to be inhibitory. If the organisms make it through this and are able to survive through the stomach acid, then you've got the bile, right, which is a little more on the alkaline side, and so that can have inhibitory effects for some of your microorganisms. We also have iron binding proteins. This will pop up in the next slide again with some of our antimicrobial peptides for this particular text. They had it kind of linked with that. But the iron binding proteins we're going to find in different fluids that are produced. And iron, why would that be important? Well, iron is going to serve as a cofactor for many enzymes for metabolism. And so when the iron is bound to the protein, it means it's not accessible. So you're reducing the accessibility of iron, and now these enzymes that require it to work properly they don't have access. So that metabolism isn't going to happen. And many of our bacteria do require that iron as a cofactor for some of their major metabolic functions. That being said, one of the ways microorganisms have gone around that is, some of your microorganisms, their proteins have changed, so they no longer require iron as that cofactor, but it may another metal ion as a cofactor instead, and therefore that would no longer be a problem for that particular pathogen. For the antimicrobial peptides, these are present on the skin, mucous membranes. We find them in the neutrophils. They act against a broad range of microbes, right? They work in several different ways, such as cytolysis. They interrupt enzyme action. Some of them actually assemble into are fibers that assemble together to create these fibrous traps that then trap bacteria, making them easier to remove from the system. The antimicrobial peptides tend to be broad spectrum, effective against bacteria in general, both gram-positive and gram-negative, as well as your fungi. So a couple examples that your text has brought up would be our bactericidins, right, produced by some of our resident microbiota. And so that's going to be found in the gastrointestinal tract, and it's going to affect the bacterial cells by disrupting their membranes. Okay, we have cathelicidins, which are produced by some of our cells from the skin, right, and that inhibits bacteria and fungi through disruption of membranes. And we have defensins, again, produced by our cells throughout the body, affecting bacterial, fungi, and even some of your viruses, again, disrupting membranes. So if you're talking about viruses, that disruption would be the envelope, right? And then dermacidin, which is going to be your sweat glands, so therefore we find them on the skin. That's going to affect bacteria and fungi, disrupt membranes, also can affect your ion channels, so that that balance between your positive charge and negatively charged ions that are moving in and out of the cell. And the histidins, which are salivary glands, disrupt intracellular function for fungi. Okay, so those are a couple. We also have all of these basically, produced both by us and or our microbiota to prevent these pathogens from the potential pathogens from establishing within the system. We also have a group of what they refer to as acute phase proteins, another class of these antimicrobial proteins that, in this case, are going to be produced not by any cells, but really produced in the liver, and then is secreted into the blood. So it's going to play a role in your inflammatory, with your inflammatory response, right? And the innate immune system. Some of these we actually will be talking about in the process as we go through this process. For instance, we are going to be talking about complement. Mannose binding lectins is one of the pathways to get the complement system activated. So that'll be actually discussed a little bit later when we go through complement system. You can see here we have two, ferritin and transferrin, both of which are those iron binding proteins. So they pull the iron, making it less accessible, so that inhibits the growth of pathogens that require it for their metabolism. We have a couple here, C-reactive protein and serum amyloid A. They actually coat the bacteria, making them easier to recognize for phagocytosis. This term, opsonization, we're going to see again, right? And that's, so I'll be talking about that later, but that's that idea of, again, promoting phagocytosis, okay? All right, and then fibrogen is going to be that one of those that is going to form these nets or traps that as it interacts with bacteria and each other in order to create that, and kind of create a larger, basically kind of a larger molecule between the fibrogen and the bacteria, which is then easier for some of our organelles to be able to remove that pathogen from the system. All right, moving to the second line of defense. The second line of defense operates, or is going to come into play after the pathogen has gained entry, okay? So the pathogen has penetrated the skin or mucous membrane, gained entrance into the body, and now we're looking at, trying to control what has already gained entrance, okay? To do this, we talked about the fact that there are chemicals that are going to come into play. There are cells, specialized cells that are going to come into play. So we're looking at composes these antimicrobial chemical cells and the combined process, the combined of these units are going to create these processes. And the processes we're going to be looking at would be the inflammatory response, phagocytosis, complement system, and interferon, as well as fever. Okay, so those are the 5 things we're going to be looking at. So many of the components that are going to play a role in this process are actually originating from the blood. So we do have to kind of talk about, the blood itself. The blood, of course, is composed of plasma and formed elements. The plasma is the liquid component of it, mostly containing water with electrolytes, dissolved gases, nutrients, and proteins. Okay? If you remove the clotting factor, which falls into that protein component, if you remove the clotting factor, that's what we refer to then as serum. Okay, so the serum is plasma, so no blood cells, just plasma, the liquid component with the clotting factors removed. Now, the plasma itself, the components of plasma, besides water is very important for metabolism in general, but besides plasma, of course, the electrolytes are going to be important in providing potential cofactors, right, needed by the cells for metabolism. The gases, depending upon whether they are exchanging gas, whether the organisms are aerobic or anaerobic. For our cells, of course, we need that oxygen coming into play. But some in the intestines is primarily anaerobic, So the presence of the gas, of bringing in those gases is important in that process potentially. It is the complement as far as nutrient, well, before I go too far further, nutrients are going to play a role in recovery and for the repair, should you have a nick or a cut, so in the inflammatory response. And then the proteins are going to include the complement proteins, and antibodies, all which are important in the immune response. Some antibodies are actually important for the third line defense, and complement is important, is part of the second line defense, okay? So those are present in the plasma. So plasma has its own function as to its importance and what it carries throughout the body. But in addition, of course, we have our formed elements. And our formed elements, in that grouping of formed elements, we have the erythrocytes, which carry the oxygen and carbon dioxide in the blood. The erythrocytes are not involved in the immune response. Okay, they're important, very important for the health of the organism, right? As in the host organism. But as far as the immune system for the organism, they are not playing a role. The platelets, though, these are involved in blood clotting. That's going to be part of that second line defense. And then we have our leukocytes. The leukocytes are our white blood cells. The leukocytes are divided into granulocytes and agranulocytes. Okay, and so we're going to go through those as far as what's what. But the leukocytes definitely are involved in the immune response. Okay, they're going to be important in this process. And then we actually are going to, in a little bit, we'll talk a little bit about how we actually look at a white blood cell count, that is the count of the leukocytes, to try to identify what type of pathogen an individual might have. Okay? So when you look at this flow chart here, of course, our stem cells are found in the bones, right? And within that we have differentiation into either the myeloid stem cell. The myeloid stem cells are the lymphoid stem cells, and we have differentiation into then either the myeloid goes into platelets, important for the immune system. Erythrocytes, that's not involved in the immune system, it's just for oxygen exchange. We have the mast cells. The mast cells are they play a big role in signaling, so they do play a role in our immune system. And actually they play a huge role if anybody has allergies. Okay, so they're going to be a big player in the allergic response. And the myeloblasts, which is going to differentiate into basophils, neutrophils, eosinophils, and monocytes, all of which are part of that second line of defense. The monocytes will differentiate into macrophages or dendritic cells, depending upon where they are located. And then we have the lymphoid stem cells. This side here, for the innate, non-specific immunity, we have our natural killer lymphocytes. So those will actually play a role in the second line of defense. The T cells and B cells, we won't talk about these until we get to the module 12 and with the specific immunity, okay, because they're going to play a role there. Now, just as a side note, so that you're aware, and I think you guys probably are, but I am talking about first line of defense, second line of defense, third line of defense, right? This is not quite like a sport where you pull upon a line of defense only after the previous one fails, right? The first line of defense actually, I'd already mentioned the dendritic cells monitor, so we have some dendritic cells that monitor those areas like the skin and the mucous membranes. And so that side of phagocytosis, part of that second line of defense, does somewhat play a role even when we're primarily focusing on the first line of defense, of getting the pathogen even entering into the body. Once the pathogen enters into the body, We're focusing right now on this module for the second line of defense, but be aware that the third line of defense is working at the same time, right? We're not waiting around, well, now the first line of defense failed, so in comes the second line of defense, the third line of defense, right? Second line failed, third comes in. That's not how it works. It's consecutive, it is happening, it's not consecutive, it's concurrently happening, okay? And throughout this presentation, as well as when I get into the next module and explain, I have to explain sections or parts. We have to realize that the parts all, again, overlap. Okay, so to try to, you can't explain all at one time. You kind of have to explain the parts first before you can explain the total. Okay? All right, so when we are looking up here, for our, so we have our granulocytes and our agranulocytes. Our granulocytes, these are recognized and referred to as granulocytes because when they are stained, they have these, these little dots within them. These are vesicles with signaling molecules already ready to go so that when the event occurs, it can then communicate with other cells in that immune response. And some of them, may even have, your lysozymes and other vesicles. So it's got a lot of vesicles present in it, which give it a granule appearance. So your granulocytes, there are three main types. Those are the basophils, eosinophils, and neutrophils. The basophils consist of, and you don't have to memorize these percentages, right? But basophils are at about 1% in the blood. So if you're doing a white blood cell count, you're looking at those ones. They all stain differently, so you can actually do a count of which type you have. And they're usually sitting, in a healthy individual, they're usually sitting about at 1% of those white blood cells. Eosinophils usually sit around 2 to 4%, okay? And then neutrophils are the ones that are highest in the blood at about 60 to 70%. The eosinophils and the neutrophils are the two here that have the ability to perform phagocytosis. Okay, so they're going to have that capability of phagocytosis. They also have that capability of diapetesis. Diapetesis is being able to cross the blood vessel into the damaged tissue area when we have vasodilation. Okay, so that's a part of the inflammatory response. The agranulocytes, there are two. They have the lymphocytes. The lymphocytes are usually at a range of about 20 to 25% in the blood, so for when you do the white blood cell count. That's in a healthy individual. The lymphocytes are primarily the B cells, T cells, but again, that also includes the natural killer lymphocytes. And then we have our monocytes, and our monocytes are usually about 3 to 8% is in a healthy individual. Okay, so again, not expecting you to remember the percentages, but I do want you to recognize what it means when they're different. So when we have an individual who is sick, and we're not 100% certain what is causing the disease state, right? So we want to kind of get a general idea. This is where they're going to do that differential white blood cell count. And with the differential white blood cell count, they can signal based upon the percentages of these different white blood cells. It can signal to us or kind of indicate to us what type of pathogen they might have. Okay, if you see an increased number of eosinophils, that usually indicates an allergic response to something, or parasitic worms, the helminths. Right, they have an infection of a parasitic worm. If you see a increase in white blood cells across the board, right, just across the board, you have a high white blood cell count, as well as your neutrophils are on the higher level. that usually indicates a bacterial disease. And if you see an increase of lymphocytes, okay, that usually indicates a viral infection. Okay, so the white blood cells that are the counts of these white blood cells and which ones you have more of can give you an indication as to which type of disease or what type of pathogen you might be looking at. Lymphocytes, these acquired ones, especially our T lymphocytes, generally linked to viral infections, right? So higher levels of those generally link to a virus. All right. Now, so those are these are the cells involved. What about some of the chemicals involved? Well, the chemicals involved would be our cytokines. Cytokines are signaling molecules, right? They are proteins that they serve for communication purposes. They are produced by various cells and then released into the environment. They can serve as informing cells, or as chemotaxis, informing cells to come to this area to do a particular area. So bringing cells into an area, they can be involved with cell proliferation. Hey, you need to increase your numbers. They can trigger cell death, so apoptosis. Okay. Or they can trigger differentiation. It depends upon the particular cytokine that is being released. The cytokines play a role in the inflammatory response, okay, also. They are going to promote that inflammatory response through releasing proteins, the cytokine proteins such as histamine, leukotrienes, prostaglandin, and bradykinin. Okay, all of which are also recognized as linking to, say, an allergic response, an allergy response, right? Many of those will cause vasodilation. They have chemotactic properties in order to then promote an activity. Now, cytokines can be chemicals that are signaling molecules that are secreted by the cell to be received by the cell. Okay, and we're going to see that in these type of cytokines are called autocrine. Their goal is we're going to see that with, say, the acquired immune response as a means of when an activated cell tells itself to produce more, clone itself, produce more copies of itself, cell proliferation, make additional copies. It can be the cytokines that are secreted by a cell that's near another cell, so they're close by, are the paracrines, and so that's a close interaction, versus the endocrine would be where you're looking at a long distance. Right, so these are going to be traveling through the circulatory or the lymphatic system to go to another location in order to signal and inform, right, that an action needs to happen by certain cells. All right. So we are now going to. We're actually in our last learning module based on the list, but it's actually pretty packed, right? Because we're going to go through phagocytosis, we're going to go through the inflammatory response, complement system, interferon, and fever. Okay, not in that order. I have my order going through it, and it's not the way it was listed. So we're going to start off with phagocytosis, though. The cells capable of phagocytosis are called phagocytes. Okay, so and there's a group of them, right? The three main types are the neutrophils, the monocytes, and the macrophages and dendritic cells, right? So really the monocytes are going to differentiate into the macrophages and dendritic cells, right? When so it starts as that, and they differentiate depending upon. When they're in the tissues, so outside of the blood, they are inside the blood they're monocytes. Outside of the blood, then they are those macrophages and or dendritic, depending upon which tissues. The phagocytes, their job, of course, is to survey tissue and blood, right? And their goal is to identify and remove any microbes, any foreign material in general, right? And they do this so it can be microbes, it can be particulate matter, it can be injured or dead cells, even host cells, even your own cells, they're going to identify and get rid of. The job itself involves ingesting and then eliminating that material, so breaking it down. And in that process, they're going to extract antigens and display them on the outside of the cell for your third line of defense, for your B cell, T cells. Okay, so that's going to be something that is another kind of not just removal, but they're going to be promoting the recognition of these antigens by our acquired immune system. Phagocytic cells have to recognize what is foreign, what are the microbes. So to do so, they have receptors on the surface of the cell. These are referred to as toll-like receptors. Toll-like receptors are, depending upon the particular receptor, they are either inserted into the outer so the membrane itself of the cell, or they might be inserted into a membrane of a vesicle within the cell. Okay, it depends upon the particular toll-like receptor. But the toll-like receptors are going to bind to those pathogen-associated molecular patterns called PAMPs. Okay, and what are the PAMPs? They are, they can be peptidoglycan. They can be the lipoteichoic acid of, say, the gram-positive cells. They might be lipid A of the gram-negative cell wall, right, the outer membrane. They can be the cell wall of yeast. They can be the flagellin, which is that protein that that the bacterial flagella is made out of. Okay, they can be double-stranded RNA. They can be single-stranded, viral DNA or RNA, methylated in such a way that it's recognized as not ours. Okay. So they interact with that. That's what makes the innate immunity able, these phagocytic cells, be able to recognize what is foreign, but not be specific, right? Because peptidoglycan is going to be exposed and present on any gram-positive cell, right? Lipoteichoic acid is going to be on any gram-positive cell, not a particular one, but any bacterial cell that's gram-positive. Lipid A is found on the outer membrane of any gram-negative cell. Okay, so the specificity is not there, but it is recognized as not belonging to us, right? Because these are molecules that we don't produce, and that's how they are going to be able to recognize them. So, what are the stages of this process? Well, the first stage, okay, is chemotaxis. And this is actually is going to be linked to, say, if you have damaged tissue. So when you have damaged tissue, your cells will start to secrete these cytokines that are attracted to chemotaxic chemicals that are going to trap the phagocytic cells. So when they're in the area where this pathogen is, they need to then adhere to it. So that's where the toll-like receptors are going to interact with that PAM. whatever it might be, okay? Once that interaction has happened, adhesion occurs, that is going to trigger endocytosis. Okay, so interaction between the PAMP and the toll-like receptor triggers endocytosis, and so you get the bacterial cell is going to be engulfed, and you'll get the formation of what we call a phagosome. The phagosome is going to be a vesicle that contains that pathogen. We then need to have the breakdown of that pathogen. So the breakdown of the pathogen means that we need that phagocytic cell, it needs the lysozyme, that kind of the stomach organelle of the cell, is going to fuse with the phagosome to create the phagolysosome. When it does so, those digestive enzymes and the low pH within the lysosome is going to transfer to that now phagolysosome, and those digestive enzymes are going to start to break down that cell. Okay, and so we get that's that maturation process and killing process. So it's ingestion is taking it in. Now we've got maturation is when the lysosome interacts with the phagosome to become the phagolysosome, and then we have killing that occurs as the cell is broken, that bacterial cell or pathogen is broken down. The last step is to eliminate the debris, okay? But what also happens inside here is we do have some of our major histocompatibility complexes that are important for the response of the acquired immune system that's going to be able to interact with pieces that might have antigenic properties. So that when this fuses with that, when the phagolysosome fuses with the cell membrane, the contents are going to be dispersed into the environment. That gets flushed with either the circulatory system or the lymphatic system, right? And the debris gets removed. And any antigenic properties, specific antigenic properties that cell might have is going to be then displayed on that phagocytic cell for our acquired immune system. Okay, so it is actually going to stimulate the adaptive immune response. All right. What else do I have? So I had talked about toll-like receptors. What else do we have? We actually, inside some of our cells, we have what we call NOD proteins. The NOD proteins are not attached to a membrane. They are actually proteins that are found in the cytoplasm, and they too bind to PAMPs. And when they bind to these pathogen-associated molecular patterns, they will trigger inflammation, apoptosis, and other of our innate immune responses. Okay, that's still kind of a newer area of that they're still researching the full function and how it works, but it does allow then for other cells, not just phagocytic cells, to be able to respond to these PAMPs that might have gained entry into the cell. All right, so this is just showing you, just showing you the examples of these toll-like receptors and interaction with the potential PAMPs that they would recognize, and upon interaction with the PAMPs, that is going to trigger a series of events in order to promote that phagocytic process. Okay, so I've already talked about this slide. This is just they showed as they are cell surface receptors that are going to recognize these specific pathogen-associated molecular patterns. that being said, I should also note, because the phagocytes also are going to be, through optimization, that process is going to be promoted by complement, okay? And so complement system is, and as well as by antibodies from the acquired immune system, these two systems, you're going to find that the phagocytic cells have cell surface receptors that recognize certain complement, so that when we get to the complement, you're going to find that some of our microorganisms will get coated with a certain structure, certain complement protein, and that serves as an additional flag that can interact with phagocytic cells to promote phagocytosis. Okay, antibodies, same thing. We have, with our antibodies, we have variable regions that recognize can then alter in order to recognize different these different antigens, but we also have constant regions. The constant regions are important because our phagocytic cells have a cell surface receptor that recognizes that constant region, right? Bind to it, and that then promotes phagocytosis of a pathogen that an antibody has recognized. So once again, this is going to be one in which I can talk about the pieces at a time, which is the only way to address this, right? But all the pieces are linked together in various ways to give us a stronger response, okay, to that particular pathogen. All right, I'm taking a 15-minute break so that I can rest my voice. So we'll give it 15 minutes, give you a chance